Hello, my name is Kareem Shaheen in period 2, and I'm going to be going over the different stages of mitosis. So I'm actually going over and labeling them as the steps of the cell cycle to include interphase, which is the first phase. Then it moves on to prophase. After that, it is metaphase. And then anaphase. And then telophase. And it all is completed with cytokinesis, which is actually a part of telophase. However, it's pretty distinct, so I'm going to be discussing it on its own. So these centromeres here, while initially somewhat, so what they do is moving into prophase from interphase, they move to opposite sides of the cell. So I'm going to be readjusting that now. And also within the nucleus itself, all the genetic information is switched out into organizing as into chromosomes and moving away from being bundled up, so that now it's two chromatids combining together, chromatids to make up each one. So now I'm going to be moving on to the next phase, being metaphase. So what happens in metaphase is the spindle fibers and microtubules come out in order to grab these the two chromatids and to separate them after they have been lined up. And that moves us on to anaphase, which is when they actually are pulled apart into the two chromatids, so that all the genetic information is separated into the two poles of the cell. So I'm going to be drawing that out right now. Okay, so this is anaphase right now when they're separated. Okay, I'm now going to be moving on to the next step of it, which is telophase. At this point, the chromosomes are at their poles and are becoming more diffuse. So what happens is now a new nuclear envelope becomes formed, and the cytoplasm begins to divide. So right now I'm drawing the nucleus of one side of the pole, and you can start to see that it's becoming more, they're becoming two individual cells, and we're almost at the end. So you can see them with their own new nucleus now as the new nuclear envelope forms, and you can see the centromeres also going to their own side. And then when it finally claps off into two individual cells, that is cytokinesis, finishing the entire process. So now I'm going to be going over specific mitotic questions. So question 1 is, if a cell contains a set of duplicated chromosomes, does it contain any more genetic information than the cell before the chromosomes are duplicated? This is not true, as the genetic information regardless is the same, considering it came from the same chromosomes and it's just being divided out, so it's the same. So question 2 is, what is the significance of the fact that chromosomes condense before they are moved? This is done in order to make sure that during the process of the movement, it doesn't get tangled. And then what happens is, for question 3, how are the chromosomes copies called sister chromatids separated from each other? So what happens is the centromere is used to pull them apart into the different poles of the cell. And for the final question is, what would happen if the sister chromatids would fail to separate? This would result in one of the cells having an extra chromosome, and then the other one would be missing a chromosome. Okay, that is all of the different parts of this mitosis, and as well some questions that generally go along with it. Thank you.